Hey guys, welcome back. We're going to start off today where we left off before the break, looking at clinical laboratory instruments, and more specifically looking at a technique called electrophoresis. So electrophoresis is a method of looking at different molecules um, based on their properties and the idea that these molecules uh, are charged when placed in a medium, and if we apply an electric field to those to that medium, those particles will move through that medium. And so electro meaning electric field, phoresis meaning the movement or the migration of those molecules through that electric field. All right, and so this is based off the idea that biological macromolecules are usually charged. All right, so therefore they will move in the presence of this electrical field, like I said. And so three big purposes for maybe wanting to carry out electrophoresis is one, to determine the number or the amount of stuff that we have and the mobility of that stuff, right, in a given sample, right? So how it is able to move, how freely is it able to move through the medium. And to obtain information about electrical double bilayers, so this is like looking at membranes that proteins might sit on, things like that. All right, and then also looking at determining the molecular weight of those proteins, or DNA, and we'll talk about each one of those in these upcoming lectures. All right. So electrophoresis is one of the most common methods, all right, for looking at separating these molecules, these particles, right? Some of the biological molecules that we're talking about here, things, I've just listed a few, proteins, carbohydrates, nucleic acids, all right, polysaccharides, peptides, amino acids, nucleosides, organic acids, small ions or cations that you might find in different biological fluids such as blood or urine. All right, electrophoresis is an analytical tool. It's very simple, inexpensive, and highly sensitive. All right, so if I were to take, a sample and place it inside of this particular medium here, any kind of place along there, all right, then depending on the charge or depending on the properties of this molecule, all right, the positively charged molecules are going to migrate towards the negatively charged electrode, right? And the negatively charged particles will migrate towards the positively charged anode, okay? And so the way that, or the rate at which these molecules will migrate towards their respective anodes are going to depend on a few things, such as the net charge of those molecules, all right, the size and the shape of those molecules, and then also the magnitude of that electric field. Some of the other things that might affect how these migrate will also be things like viscosity or the temperature in the actual medium. We can mathematically describe this through a couple of equations. One's the velocity of migration, all right, and this allows us to describe that rate of migration, all right, through the medium, all right, described as U equals Z times E divided by F, all right. So ZE is actually the net charge of all of a particular molecule, all right, and so Z is unitless, all right, it's just the number of charges, all right, E is just a constant that we use and measured in coulombs, all right. E is going to be the magnitude of that electric field, all right, measured in volt per meter, all right, and then F is a frictional coefficient that actually helps describe the actual size and shape of the molecules, and that's going to be in kilograms per second. Another important equation is the electrophoretic mobility. All right, so this tells us really how freely can these molecules move about this particular electric field. All right, and so the velocity of migration and electrophoretic mobility can be used to measure, right, the charge of a particle, right, in say a neutral or non-conducting medium. And this is actually how the charge of the electron was originally measured. Okay, and so, however, though, biological macromolecules are usually found in aqueous solution, right? So these sit in our body, right? And so they're going to be in place with other ions, buffers, and so on. Therefore, charged molecules are surrounded by what we call like an atmosphere of different ions. So we want to be able to separate those molecules from all those other ions as well. All right. So some of the components, all right, in an instrument that we would use for electrophoresis are going to be things like the electrical power. This is the driving force. This is what's going to give us our charge to the medium. Okay. Um, the gel, right, is the medium that we're going to use to actually separate the different molecules, and we'll talk about that. There's a buffer, all right, and this is a support medium, all right, which helps maintain the pH or the strength or maintain the viability of the molecule that we're looking at. All right, and obviously the sample that we want to look at, the molecule of interest, all right, and then also we're going to have some kind of detecting system, either quantifying or qualifying the actual results from our electrophoresis. So there's a couple different types of electrophoresis to be aware of. There's really two different modes and methods that we talk about, either free electrophoresis or zone electrophoresis. And mainly we're going to be focusing on different types of zone electrophoresis. All right, and so here we have just an old standard paper electrophoresis or a cellulose acetate electrophoresis, or mostly we're going to be talking about gel electrophoresis. So just to give you an idea of the different types of techniques that are out there that you might hear about as well. But we'll mainly be focused on gel electrophoresis. So, like I said, gel electrophoresis, nearly all electrophoresis of biological macromolecules are going to be done in gels. All right? And gels allow us to separate molecules according to their charge or size or even their shape, depending on what we want to try to, how we want to try to separate it. Okay. There's a couple different types of gel we'll discuss as well. And in general, all right, we can get qualitative measurements using an agarose gel, all right, and quantitative using different techniques such as page, which we'll talk about in upcoming lectures as well. So really, in a gel electrophoresis, the primary medium is going to be some type of polymer, right? Some kind of gel polymer. The two type are an agarose, right? So these gels are 3D polymer networks, okay, that we disperse into a particular solvent, all right? And agarose consists of an aqueous medium, all right, and a polysaccharide extract, all right? And the polyacrylamide, all right, is going to consist of an aqueous medium and a copolymer of acrylamide, all right? And we can see some differences as well here in the particular gels, all right? Agarose we might use to separate more larger molecules, where polyacrylamide might be used for small molecules, all right, such as protein, things like that, all right. You can stain both these gels either before or after the actual gel is done. And these gels contribute also some additional factors that will affect that electrophoretic mobility, all right, how freely these molecules can move about. All right. One is the gel imposes some additional friction, all right, some resistance to the molecule as they move through these 3D structures, these different pores, right? And the molecule also interact with different charge groups on the actual network of these 3D structures, right? And then finally, the molecule or some of the pores in the gel will be too small for the molecules to enter, right? So that's how they get separated. And all these factors end up improving the resolution of our electrophoretic technique. Like I said, the medium is going to sit in a different aqueous buffer as well, or in some types of buffer. All right, and here are just a list of some of the different types of buffer that you may see. All right, and these buffers allow us, they carry the current. All right, so they're going to carry that that charge between the positive and negative electrodes. All right, and also the buffer helps protect the samples during electrophoresis. All right. And so if we're looking, the two big things we're going to be looking at are like DNA and proteins for electrophoresis. And if we're doing some type of DNA separation, all right, we might use like a tris borate, all right, or a tris acetate, or a tris phosphate as a particular buffer. And for proteins, a TBE buffer is can be typically used. All right. All right, so we talked about the gel, the medium, and the buffer that we're going to need to perform electrophoresis. But let's talk about some of the equipment that we're going to need as well. First thing we're going to need is some type of mold. And there's two types of molds. There's a horizontal mold or a submarine mold. All right, and for a horizontal mold, all right, the mold sits flat on the table. All right, and our electrodes come in on the side here, okay, our negative electrode and our positive electrode. Our buffer solution, all right, will sit inside of our mold, cover our mold completely, all right, and then our gel or our medium will sit inside of the mold as well. And so then as our current is applied to this particular mold from the negative to the positive, our molecules are going to travel, especially for DNA, travel from the negative electrode to the positive electrode, right? And so they will migrate down the gel like so. All right. Some of the things that horizontal electrophoresis might be used for, things like immunoelectrophoresis, isoelectric focusing, which we'll discuss more of, DNA separation, or RNA separation. There's also a vertical gel mold, right, which is the same thing except flipped vertically, right? These are typically used for the separation of proteins, right? And so here our mold will sit vertical and our gel will also sit vertical. So you can see here our buffer solution, all right, will fill up about usually fill it up about 3/4 of the way, all right, and then our gel will sit into the solution as well, all right, and then our electrodes, all right, our negative electrode is placed up here at the top, our positive electrode down at the bottom. And the same idea, all right, as we apply the current, our molecules will travel from the negative electrode to the positive electrode, and so as our gel runs, all right, the molecules will travel from the negative down to the positive electrode. Combs, these are used to put the different wells inside of the gel and allow us to load our samples into the gel. All right, two kinds of combs. We have a regular comb, all right, in which we have these little square blocks. All right, they're going to be created, all right, for us to be able to put our sample into. A houndstooth comb is a little bit the same, except gives us a little bit of a smaller loading well in order to, or a little bit more of a precise loading well, so that we can load even smaller molecules into our gel. So let's talk a little bit about how we can use electrophoresis to separate or look at DNA. So gel electrophoresis is a technique, all right, and provides a rapid and efficient or an effective method, all right, to determine the sequence of a, say, like a single strand of DNA. And so why would we want to do this? Well, perhaps we want to do some gene identification, DNA sequencing, all right. Maybe we want to diagnose different genetic diseases or look at gene expression, all right, in the lab. And then for forensics or paternity testing as well, we may want to separate different DNA. We'll talk about that. So the basic principle behind this is that DNA is negatively charged, right, due to its sugar phosphate backbone, all right, and from this, DNA will be attracted, all right, to the positive anode, or the red anode, and then also repelled by the black cathode, all right. So because of the negative charge, it's going to want to migrate to that positive electrode, all right. And so if we fill our well like this, all right, then we'll see that the larger molecules are going to take longer to migrate through the gel, so they're going to get stuck back towards the back, towards that negative electrode. But the smaller ones are going to move more rapidly and move quicker through the gel. And even smaller ones will move even more rapidly towards that positive electrode. And so the way that we do this is that we use, we take the DNA and we use these restrictive enzymes to cut up the DNA into these smaller fragments, or different size fragments. And so the size of the DNA fragments is going to affect how far it will travel. So remember I said that the gel has these pores, these 3D structures, these pores. And so the molecules are going to migrate through those pores. And the longer molecules, all right, are going to be stuck back towards this negatively charged electrode, all right? But the DNA fragments that have been cut up smaller will migrate through those pores more easily and migrate down towards the positive end, okay? And so here's another little graphic for you guys. All right, kind of shows the whole process. All right, so we take a mixture, all right, of DNA, all right, of different sizes. So we've introduced that restriction enzyme, cut up those DNA fragments, okay, and so we have these samples of molecules of different size DNA. These are then placed into our wells, all right, and then we turn on our power source, all right, so our negatively charged and our positive charge. These molecules are going to migrate, all right, in the gel towards the positive end, all right, and then when we have completed our gel, our longer molecules, all right, are going to be left up here towards the negative, and the smaller ones are going to be down here towards the positive end. All right, and so we might end up with a gel that looks something like this when everything's said and done, all right, where we have different samples of DNA, all right, and then depending on what we want to do with that, how we want to quantify that, all right, we can do different things. The other thing we're going to need to do is stain, all right, this DNA, all right, so that we can view it, all right, and usually we do that with some type of this ethidium bromide, all right, which will bind to the DNA and allow it to fluoresce, all right, under particular UV light. And so that we take our gel, place it under the UV light, and because of that ethidium bromide, right, it's going to make these stains kind of glow. It's kind of neat looking. All right, and then this is another graph showing you that how DNA will migrate. All right, so it's inversely proportional to the log of their molecular weight. So so if this is a graph of our molecular weight, all right, or base pairs, all right, then the distance that they will move, we see that the DNA fragments with the larger base pairs, right, don't move as far as, say, the smaller DNA fragments with the less amount of base pairs. All right. So if I were to do a log transform of this particular graph, all right, we would see that we get a that's proportional, all right, to the log transform of the base pairs. And so when we're trying to detect or look at DNA fragments, these different sizes of DNA fragments, we typically use what is called a ladder or standard, right? And so this is a known DNA ladder or standard, right? That usually gets placed in the first well or last well, depending on how you're doing it. And then those fragments will then separate out to known sizes. Right? For this particular ladder, this happens to be, 1,200 base pairs here, all the way down to 100 base pairs at the bottom. Right? And so then we can place our samples into the other wells, all right, and then run them and compare them to our DNA standard and determine what the molecular size of each of those particular runs are. This is another slide showing you guys different DNA stands. So we talked about lithium bromide, which we need to use to view these under ultraviolet or UV light. All right, looks something like that. And then there's a bromophenol blue, all right, which we can stain without having to really need the ultraviolet light. All right, and this stain will allow us to view it under just a typical normal light source. So two different ways to stain our DNA. All right, so one of the things we might want to do with DNA using electrophoresis is look at the coding for individuals and get their fingerprinting. All right, and so although all humans have similar sequences, right, in their DNA, each person's DNA pattern differs subtly, very differently. And one way is that sections of their DNA, which we'll call junk, and this is just stuff that doesn't code for proteins, all right, are made up of different size patterns, all right. And so may have a sequence that looks something like this below, all right, where in the green area, if you can see that, this green area, all right, is a coded area of different junk here, all right. And so this person here, all right, and their DNA strand, they might have a length of these different repeats here, all right. But another person might have a different size of repeated patterns. And so these little subtle differences in the repeats of their patterns can distinguish between two people. All right, and so DNA samples, for instance, obtained either from like the skin, the blood, uh, semen, hair, all right, or any of that that might be like say left at a crime scene, for example, can be compared with the DNA from a suspect to see if they like match. All right, so we can take the fact that each of us have um, different links of non-coding regions in our DNA, and we can develop fingerprint profiles for different people. All right, and so we can look at the patterns and develop these different fingerprints. All right, and so I might have this one person. I take a DNA swab from them. All right, I get a DNA profile that looks something like this. All right, this is their DNA strand, and let's say I want to cut this DNA, and I'm going to cut it at the sites of guanine. All right, so where guanine repeats. All right, and so if I do that, I will end up with three different strands or three different uh, segments of DNA, right? So here's the first, here's the second, here's the third, all right? And so I've cut this DNA and I'm going to place it in the gel, all right? And if I place it in the gel, all right, the highlighted DNA fragment, all right, is going to migrate to say this place in the gel, all right? And then the other two look a little bit smaller, all right? So they will probably migrate a little bit further based on their size, all right? So they're going to migrate a little bit further into the gel towards the positive end, all right? And so now that I've got this profile for, say, person one, all right, I can do the same thing for another person, right? And so now I can take the second person, I can do the same thing. I can do cuts on guanine in this particular case, all right, and I can see that I get different amounts of uncoded regions than I did in person one, all right. So person two has longer uncoded regions than person or person two has longer coded regions than person one. All right. And so if I do this, I'm going to end up with the same three fragments, all right, but they are going to be of different sizes. All right. So see if I put this in the gel and run it, all right, this section of code, all right, that's highlighted here, all right, is going to be longer because of those longer uncoded regions. So it's not going to travel as far, all right, as the other, the shorter non-coded region. And then the other two, the same length, are going to end up in about the same place. And so this difference, all right, between this uncoded region here and this uncoded region here can help us distinguish between this person. Maybe person one was, you know, some DNA that was particularly found at a crime scene, all right, and then maybe person two was a suspect that they had, all right, and so if they run this, they can see that person two obviously didn't match the evidence that they collected at the crime scene. Another thing we can do by analyzing DNA through electrophoresis is we can do evolutionary relationships. So we can compare DNA samples from different organisms and tell how similar, all right, they are to one another. So here I might have an example of looking at DNA from 5 different species or different organisms, all right, and so I can get a DNA fingerprint or DNA profile from species 1, species 2, species 3, 4, and 5. All right, and then I can create a evolutionary tree. All right, and determine that species 1 and 2 are pretty similar. All right, in their particular DNA profiles. All right. 3 and 4 seem to be very similar, all right, in their DNA profiles. And then 5 seems to be kind of not too relative or too related to any one of those, because it kind of sits on its own branch. And if I were to actually look at the different, if I were to classify these, if I knew the classification of these DNA samples, then I could look at how similar they are. So turtle is very similar to snake, a rat similar to squirrel, and a fruit fly sitting on its own here in this instance. So another reason we may want to use DNA electrophoresis is to diagnose different medical issues that patients may have. A couple other examples are like looking at patients that have sickle cell disease, all right, compared to those who have normal blood cells, all right. Another one is for looking at Huntington's disease in this particular case. All right, so if I know of the particular allele or the region of DNA that codes for the protein Huntington's disease, I can look at that particular allele, and I can look at a chromosome that has a normal allele, all right, such as this, and then I can look at that same allele on a particular patient that I suspect might have Huntington's disease. And so if I can do a DNA profile on just these regions of DNA, all right, I can see that if I were to split these alleles, all right, I would get a profile for this particular section of DNA. It looks something like this in the normal profile, right? And then if I look at the patient that we're suspecting Huntington's disease, I can see that it should look like allele 1, all right, but it doesn't. It looks totally different. And so if I know what the profile of, say, Huntington's disease looks like, I can compare that as well. So we talked about this a little bit, but we'll just drive this concept home. We can also use DNA electrophoresis and forensics to look at the DNA of a crime scene with different suspects and a particular victim. So if we have a crime scene sample that was collected and we run a electrophoresis on this, we get a profile that looks something like similar. All right, and then maybe we have a victim as well, and her DNA profile or his DNA profile looks like such. All right, and we want to compare this across the 3 particular suspects that we have for this crime. All right, so we run suspect 1, suspect 2, and suspect 3 in our gel. All right, and then we can compare all 3 suspects to our particular crime scene sample. All right, and here we see suspect 2 is the most similar to the crime scene sample because we see that their allele or their DNA segments, right, match up with one another and are the same size. So very safe to say that we can that these two this suspect 2 and the samples collected at the crime scene are the same. So we can also use this to do paternity testing and determine the father of a particular child is. So imagine we have a DNA profile from mom that looks something like this, all right? And we get a DNA profile from the child that looks something like this. We want to determine who the father is. So we take a DNA swab, we get a DNA profile of father 1, and we get a DNA profile of father 2, and we can see that the profile for father 2 is way more similar to the one to the child than father 1. So it's safe to say that father 2 is going to have to pay some child support. All right, I'm gonna stop there today. That'll end this short video of introducing you guys to electrophoresis and how to use DNA electrophoresis and some of the different applications there. The next slide is going to have a short video for you guys to watch. I want you guys to take a look at that. It's a lady from North Carolina. She's going to introduce you guys to or talk to you guys about how I actually set up a DNA electrophoresis and run that. All right, and so take a look at that. If you have any questions, get in touch with me, send me an email, or next Monday we'll get in touch, and we'll have some office hours for you guys then too. So I'm signing off, and I will see you guys next time.
I have here this lovely little gel mold, and I'm going to seal the ends of this mold with just regular old masking tape. Mind you, it has been wiped down with ethanol. So we're going to push down just like a Christmas present. We're going to push the ends down and push to make sure that when we pour our agarose in here, that it doesn't leak. And we'll do the same thing to the other end. So that should do it. Now, a lot of people put their comb in after they pour their gel, but frankly, I've never seen any difference. I've never seen, for me, any bubbling around the teeth of this comb. So I just usually put the comb in immediately. Now, we're going to make the regular agarose gel. So I'm just going to move this out of the way for a minute. And I have here in this little weigh boat about 0.32 grams of regular agarose. You can't really see it. It's very pale, and there's not a lot of it because I'm making a 0.8% regular agarose gel. So I have here a 250 milliliter Erlenmeyer flask. I'm going to pour in the agarose into this Erlenmeyer flask. And I have my 1 times tris borate EDTA buffer that I'm going to put into the flask. And I'm also going to wash out the weigh boat with the buffer because, of course, some of the agarose has stuck to the weigh boat. There we go. We have to melt that agarose, so we're going to take it to a microwave. We're going to microwave it on high for about 40 seconds till it boils, and then we're going to come back and pour the gel. Okay, so we've melted the agarose in the microwave, let it come to a boil, and now we're going to let it cool down, and we're going to put a magnetic stir bar that's been sterilized into this flask. And then we're going to turn the stir plate onto its lowest, lowest setting. And it takes 2 or 3 minutes for that to cool down. And while it's cooling down, we're going to set the pipette. We're going to put in 64 microliters of loading dye. So nice and easy, pre-wet. And it's cool enough, still very warm to the touch. We don't want it to solidify in the flask. And there's the 64 microliters of the dye that will help us visualize the bands of DNA. Now that looks evenly dispersed, that dye. So we're going to take a couple of large magnetic stir bars to anchor the small one that's inside. And now we're going to slowly pour this agarose gel into the mold. And that'll take 20 or 30 minutes to set. So this 0.8% regular agarose gel took 20 or 30 minutes to set or harden. And I've just put it down into the gel rig. The rig has the same buffer that we made the gel with, the 1 times TBE buffer. This has been sitting in there for a couple of minutes because we want the buffer to be able to get around the teeth of the comb so we can easily extract the comb. And then after we do, We're going to put in the DNA that we digested overnight using restriction enzymes, and then we're going to run it. Make sure that you're anchoring, holding down the mold. We're going to take this comb out. This is our DNA standard, or ladder, and I'm putting in 10 microliters of ladder. And here we go, there's the well. Nice and slow into the well and down into the second stop. There. And release the micropipette plunger. And now we're going to get a fresh tip for every sample. This DNA was digested with only one enzyme. So we know where this enzyme cleaves the DNA. This enzyme is PST1. And we're going to load that into the second well. Slowly. There we go. Excellent. This third vial was digested with PST1 and a second restriction endonuclease. So we're going to have DNA of different sizes. This has been digested with 3 restriction enzymes. So we'll have multiple bands appear in this well. Down into the well, and voila. We're ready to put the leads on. Black to black and red to red. This end is the negative end, and this is the red is the positive end. I want to make sure that we do the same in the front to connect those leads. I'm going to run this gel at about between 100 and 130 volts. And when I've set the voltage, I'm going to hit the run button. And there we go. Now this can run between 45 and 60 minutes. Of course, the smaller fragments of DNA, the smaller bands or fragments, will migrate much more quickly through the gel, and they'll be closer to the end of the gel, whereas the larger fragments of DNA will not be able to migrate as far in the gel because of their mass or size. And when that's finished running, you'll see that a small band of bromophenol blue will be near the end of the gel. That's when we're going to turn it off. And then we're going to look at our bands on the light box. So we have run this 0.8% agarose gel at 130 volts for 45 minutes. And we turned it off, and we stained this with a small amount of dye for 20 minutes. And then we destained it, rinsed it in ultra-pure water for another 20 minutes. And you can see now we're going to look on the light box. Slide this right off onto the light box so that we can look at the bands. And you can see here was our DNA ladder, the standards of known sizes. And look at the banding patterns that we have with these different enzymes, the banding patterns of DNA. So we can now tell the sizes, the unknown sizes of DNA by comparing to the standards. And this is very helpful to have, very important information to help characterize any novel DNA that we may be examining.